Just noticed the next three speakers, we might call this the Renaissance approach to World Trade Center research, all from Stony Brook. But we'll start with Dr. Vasca. He's trained in experimental nuclear physics at Stony Brook. He subsequently transitioned into medical imaging technology development, working in industry, followed by the PET imaging group at Brookhaven National Laboratories. Since 2001, he's been professor of biomedical engineering and radiology at Stony Brook, and currently research focus on PET imaging technology development and its many applications.
This work is from a large team of people. These imaging studies take quite a bit of help. So Minos, who you heard from this morning, Jun Zhao is our doctoral student in biomedical engineering who's done a lot of this analysis, and the rest of this team, and of course Sean and Ben, Sean Clouston and Ben Luft from Stony Brook, who have organized this particular study. So disclosures, we do have a couple of co-investigators, Dr. Gandhi from Sinai, Dr. Franceschi from Stony Brook, who have served as consultants to companies that make imaging biomarkers as well as pharmaceutical therapies in this Alzheimer's space, which we're talking about, but they have not been primarily responsible for the execution of this study, and no other authors have conflicts to disclose. So the motivation is similar to what you've heard earlier this morning. The responders have greater cognitive impairment than others who have not been exposed for their age. And the question again is it related perhaps to Alzheimer's disease? And you heard this morning about various biomarkers that we've used. And just to dig in a little bit more in these biomarkers, beta amyloid protein is one of the earliest biomarkers. It starts out in the brain as a soluble protein, and in Alzheimer's disease, the levels of it build up. It takes a fibrillar form, which then aggregates into plaques, and this is done partly by microglia, which are the resident immune cells of the brain. And those plaques are what we see post-mortem in brain slices. It's sort of the classic hallmark of Alzheimer's disease, these amyloid plaques. So they're known to be toxic. They're not necessarily causative of Alzheimer's disease. There's a little bit of debate in the community. Everyone who's been clinically diagnosed with Alzheimer's disease does not necessarily have, most do, but does not necessarily have amyloid plaques, and so there's a push in the research community to define Alzheimer's disease as requiring these amyloid plaques, and that the people who have Alzheimer's disease without them maybe have some other type of disorder, which is not Alzheimer's disease. But that's all sort of a little bit of an academic debate. Clearly, beta amyloid is central to the Alzheimer's disease processes. So this plot here, just pulled out of a particular paper. There's been many papers that show this. If you look at does this work? Yeah. It's a plot of the biomarker magnitude, where the normal is down here and abnormal is up here. And then these are going through time, through the clinical, the stages of the disease. So preclinical, which is before there are symptoms, mild cognitive impairment, and dementia. And these are the different biomarkers. So amyloid is in red here. So you see it does rise first, and this is this straight line here. Sorry, I can't move this mouse well. Is sort of like the level of detection, supposed to indicate that. So amyloid rises first. And then you have the yellow one here is synaptic dysfunction. So the neurons start to not behave as functionally well as they did before. They take up less glucose, for example. And then you have this tau protein starting to rise, and now we're getting to this range where we're starting to see symptoms. So tau is much more related to symptoms, and if you just have amyloid, many people consider that sort of preclinical disease. No symptoms, but you have that amyloid, a sort of preclinical disease. A good place to sort of start therapies, perhaps. And then later on, you get brain structure changes, like in MRI. Then you actually start to see, like, the cortical thickness actually starts to get thinner. The cortex actually starts to get thinner. And then you get more and more of these cognitive effects. So you did hear this morning that you can measure beta amyloid in the blood, but it's a little bit counterintuitive. So in Alzheimer's disease, there's actually lower levels of beta amyloid. There's higher levels in the brain, but there's actually lower levels in the blood, and that suggests that it's some problem with clearance of this protein from the brain. So if it can't clear well enough, you end up with buildup in the brain and less in the blood. So it's a good marker. It's a useful marker. It's not quite as direct of a marker as looking at it directly in the brain. So that's what we're doing in this study. We're using PET imaging, which is certainly not as easy as taking a blood sample or cheap, but it does give you a lot more information. So PET, positron emission tomography, we label a radio tracer, inject it, it goes through the blood, it gets through the blood-brain barrier, binds, it's designed to bind to these amyloid plaques, and then we measure how much tracer went where with the PET scanner in a quantitative way. So we get more information. You know, we don't just get a single number from the blood, for example, we get the whole distribution in the brain, and it's also in a quantitative way. So PET gives you the actual amount of not just a pretty picture, but it tells you the amount on an absolute scale. So the motivation, additionally, is to look at, as also Sean mentioned this morning, the potential involvement of the olfactory. So we know that toxins can enter the brain through, sort of indirectly through either the lungs or the gut. They may be getting into the blood and then passing the blood-brain barrier and getting into the brain that way. But there's also this sort of direct route through the olfactory system, the olfactory nerves, right in the nasal cavity. It has been shown in some cases to be a route of entry into the brain. You may ask how does amyloid relate to this? There's also evidence, as Sean also pointed out, that amyloid is considered by some studies that as a response to viral infection, and so there's this viral hypothesis of Alzheimer's disease, which is not globally accepted, but there's evidence for it, that Alzheimer's starts through some type of viral infection, and the amyloidosis is sort of a response to that, and it sort of gets out of hand and sort of kicks off the process. So there is some possibility that the toxins that are getting into the brain have a similar type of response, although there's not a lot of direct evidence yet in the literature, I think, that I've seen to suggest that. But we're getting these images, and we can look here and see if we see something. We also want to investigate the involvement of previously identified World Trade Center regions. So we had done previous MRI studies where we've seen irregularities in different brain regions. And so we just combined all of those into sort of a big meta region and said, maybe, do we see irregularities in those particular ones? So the methods, we have responders with and without cognitive impairment. The inclusion criteria, I think, are the same as what Frank showed this morning. MoCA greater than 26 for the unimpaired, and then less than 22 for the impaired. We did simultaneous PET and MRI brain scans, which is kind of a cool scanner that does them both at the same time. It's much more convenient. The scans are not particularly fun, but these are about an hour. We can do PET in more like 20 minutes if we just want the PET part. But we also require a lot of MRI scans at the same time. We use a couple different measures. One is called the centiloid scale, which is this funny name. It's a way there's multiple different amyloid PET tracers out there, and they all have sort of different sensitivities and so on. So this is a way to try to normalize all PET tracers. So if someone does a study with one tracer and then someone else does another study with another tracer, we can normalize them on this 0 to 100 scale, where 0 is normal, healthy controls, and then 100 is full-on Alzheimer's disease. So people usually fall somewhere in the middle of that. We also do regional analysis. I tried to show that here. It's a little confusing. This is colored regions overlaid on top of a PET scan. So the PET scan is in rainbow scale, and these colors are also kind of rainbow, so it's a little confusing. But this is just to show you that we have automated ways to automatically extract the PET signal from about 100 regions or so throughout the brain. We can compare region by region as well. And then we also did, not really the focus so much of this particular study, but we measured the cortical thickness with MRI, and then something called mean diffusivity, which is similar to what the free water fraction that Minos was talking about this morning, but also, cortical thickness lower is generally interpreted as worse. And that's a fairly direct measure of neurodegeneration because your cortex is actually getting thinner. Mean diffusivity is a little bit more indirect, a little bit harder to interpret, but higher values, as with the free water fraction, tend to be associated with disease states. So the radio tracer that we used is this one called fluor-beta-ben. It's labeled with F18. It's FDA approved. The radiation dose in these scans is quite low. It's much less than you would get in a year just from being exposed to natural background radiation. And the physicist in me always wants me to point out that, you know, PET is extremely sensitive. And one way you can visualize that is, you know, the decay of the F18 that's attached to a single molecule of this fluor-beta-ben gives us a unique count in our PET detectors. So every decay, not every decay, we miss, we maybe only collect a few percent, but Avogadro's number is pretty darn big, so we don't need a lot of grams of tracer to get a lot of decays in our scans. And we typically inject a microgram or even less of total amount of tracer into the body. So the doses are pretty low. So the results, we were able to recruit 18 impaired, 18 unimpaired, and here's all the demographics, if you can read it. Tried to match on body mass index, race, education, and so on. You see their average exposure was about 6 months in both groups, although it ranged from 0 to almost a year. And of course, so there's no significant differences, at least for what that's worth, in all these categories, and there is differences in other measures of cognition, which are in this cog state here. So we would expect that. So the typical distribution in PET looks like this. This is 3 slices through the brain of one of our responders. The red is higher uptake of the tracer, the blue is lower. And again, this is PET, so it's quantitative, so that red actually can be translated to a number, and that number represents an actual concentration in grams per milliliter of the tracer. But you could call it a bug or maybe a feature of this tracer. Actually, all amyloid tracers have a lot of uptake in the white matter, and it's not binding to amyloid plaques. So there's something about designing a tracer that binds to plaques that will also bind to white matter. So you can't look in white matter, and radiologists know this when they read these images. So what you're looking for is the uptake in the cortical band, cortical ribbon that goes around the white matter. And so again, radiologists are trained to look for this. In this particular subject, there is not a lot of uptake in that cortical band, and a radiologist would read this as negative. And the regions that we use are mostly gray matter regions to do our analysis. So we have to avoid white matter for this particular tracer. Other tracers, well, actually, all amyloid tracers are like this. Tracers for other things don't have these problems, but these do. So the results, the centiloid value, again, it was at 0 to 100 scale. It looks in certain regions of the brain that are known to be affected by Alzheimer's disease, so it doesn't look in the whole brain. And if you compare the unimpaired group to the cognitively impaired group, it is a little bit higher in the impaired group, but it's not statistically, you can see that, significant. And the other thing to realize is that these numbers are really low. This is only 0 to 20 here. So the scale goes to 100. These numbers are actually a little bit negative, which could be a bias in the analysis, but they're small. Okay, so this is not consistent. Either one of these groups is not consistent with Alzheimer's disease in terms of this particular measure. Then we looked in this meta ROI, which I said we put together the various regions that we saw in previous scans, MRI scans, into one big region, and they were affected in MRI, and we see that, okay, the unimpaired, there's more uptake of this tracer in the cognitively impaired, and it's now significant. So maybe amyloid is involved somehow, even though doesn't seem to be involved in the Alzheimer's sort of sense. So we also looked voxel-wise. There's different ways to do these PET analysis. So the voxels in PET are about a cubic millimeter or so, and so there's thousands of them, and we can just compare voxel. We put the images all in a standard space, and we compare the groups on a voxel by voxel basis. So we have thousands and thousands of voxels, so we have to do some type of multiple comparisons correction, or else we'll get a lot of false positives because we have thousands and thousands of hypotheses, essentially. So after false discovery rate correction, we found significant clusters. This is the PET. This is just showing the significant clusters over a cortical kind of image here. And you can see there's frontal regions near the orbital frontal cortex where we sort of expected to see if our hypothesis was correct about toxins coming in through the olfactory system and some other regions. Then we looked at olfactory uptake, which is, again, it's mostly the orbital frontal cortex, which is right above the olfactory bulb. It's very adjacent to the olfactory bulb. And with exposure duration over all the subjects, the unimpaired as well as the impaired, and we found a correlation with a reasonable correlation that's statistically significant. So higher exposure duration means more amyloid in this particular region. And we also explored some other regional associations. So if we assume that it started in the olfactory region and then went to neighboring regions, we just did a correlation. So this is all cross-sectional, so we can't really say that things are changing over time or anything. But there is an association, it's consistent, that the regions that are closest to the olfactory are also correlate with the regions that are a little bit farther away with high correlation. And then also that meta region that I was talking about correlates with the olfactory as well. So it does seem like amyloid is associated with this process. And lastly, we correlated our florbetaben, our PET tracer, with MRI structural measures. Like I said before, cortical thickness measured in millimeters, and this mean diffusivity property. And we can see that the direction is pretty consistent, right? So the correlations are positive for this one, which means more amyloid means more diffusivity, and that's generally interpreted as a bad thing in MRI. So we see positive correlations with these MRI measures across all these different regions, varying levels of association and statistical significance, but all in the expected direction. And then similarly with cortical thickness, but it's the opposite correlation there, because thinner, we would expect more amyloid or more disease to associate with thinner cortex, and so we get a lot of negative correlations here. One of them passes multiple comparisons here. So the impact, more of a summary, I guess, is that the amyloid measurements that we've made in the brain are not really consistent with Alzheimer's disease, at least at this point in their trajectory. But it does seem to correlate with overall cognitive measures, like the cognitively impaired are a little bit higher in certain regions. The regional changes include those near the olfactory bulb tract and could be spreading, but we don't have direct evidence of that because we only have cross-sectional data. And there is some literature to support for amyloid as an immune-like response to brain insults. Not so much, I think, for toxic dust type things, but there is some support for that type of thinking. And an open question is tau involved also? And one thing to keep in mind is that we always talk about amyloid and tau in terms of Alzheimer's disease, but they are also related to other neurodegenerative diseases. So looking at these things can be important, not just for Alzheimer's disease, but maybe identifying other ones, or this could be yet another one. They're involved in a lot of neurodegenerative processes in general. So for future research needs, if we really want to know if the plaques are actually spreading, we need longitudinal studies, as you've heard before in previous talks. Is amyloid part of an immune response? We can use MR measures, like we talked about before, free water fraction and so on. They tend to be less specific and a little bit harder to interpret. Different things can change the levels of these MR markers. That tends to be more specific, harder to do for sure, more expensive, experiments, but they're much more specific, right? If you see an increase in the PET signal, you know that there's more protein that you're looking at, or in this case, We have other markers actually for the one that was mentioned this morning. We looked at translocator protein, which is a marker of activated microglia. But we're also developing actually at Stony Brook right now, a marker for cyclooxygenase enzymes, which is another marker of inflammation. So there's many things to choose from in terms of looking at inflammation, if that's what we want to do. And then there's the question of anti-amyloid treatments, right? So there's at least one right now on the market, which is shown to actually reduce, using PET scans, it reduces the levels of amyloid plaques. It doesn't cure the disease, of course. It slows cognitive decline in a modest way in Alzheimer's disease. We probably don't seem to have Alzheimer's disease here, so what would it do in our population? Not really sure. You have to very carefully consider that. There are side effects, for sure. It's an off-label use, for sure. And there are side effects called ARIA, amyloid-related imaging abnormalities, which you need MRI scans on a regular basis to make sure you don't have microbleeds from this. So it definitely has to be considered carefully. So before I finish, I do want to acknowledge the funding and also the Stony Brook PET Imaging Corps. These are very dedicated people who get up at 5 o'clock in the morning to turn on the particle accelerator, make the radioisotope, do the chemistry, do all the QC, get it all done, and then injected safely into people every single day. So thanks very much.
Questions?
Yeah, this is Yongzhao Shao from NYU. Great talk. You mentioned in the motivation part, when you talk about Dr. Clausten's diagrams, you mentioned something about viral pathway or something. Are you indicating, does it imply the viral also cause amyloid or some epigenetic pathway?
We haven't studied this. This is in the literature. There's some evidence in animal studies to suggest, like I think Sean pointed it out, that if you have an animal knockout that has no amyloid, if you give it an infection, HIV or whatever it was, they tend to live longer. So there's evidence like that. It's not super strong, but there is evidence.
I see.
Of the viral. Yeah.
When you say the centiloid analysis data, inconsistent with the AD, with Alzheimer's, so you are saying it doesn't fit the pattern, or in other words, what's the basis for the conclusion?
Well, so normally Alzheimer's disease is 100 on the centiloid scale, and our numbers are very close to 0. So just for that reason. It's not definitive, right? But most Alzheimer's cases have much higher levels of amyloid, centiloid.
Sure, understand. Thank you.
Other questions? And thank you for anticipating my question. I appreciate that. With the FDA, do you think that'll go anywhere as a clinical? I mean, is it in wide practice, the anti-inflammatories?
Oh, anti-inflammatories. That's another potential. Yeah. So I brought up the anti-amyloid treatments, but the inflammation is something we also have to, I think, nail down probably a little bit better before we go ahead with that. But we do have these tracers that can answer that.
Thank you. Our next speaker is Laura Sampson, assistant professor in the program in public health at Department of Family, Population, and Preventive Medicine at Stony Brook. She studies the mental and physical health effects of stress, trauma, depression, and post-traumatic stress disorder, especially as they relate to sleep, aging, cardiovascular disease. She received her BA in statistics from Harvard and her PhD in Epi from Boston University School of Public Health. She completed her postdoc training at Harvard in the School of Public Health. Welcome.
Hey, thank you so much for that introduction, and thank you for having me here today. I am relatively new to this project, started working on it a few months ago, so I'm excited to be here and to share preliminary analyses on post-traumatic stress disorder and physical function over time among responders. So I thought it might be helpful to start with just a review of some of the prior studies, both in our cohort and in others, just to sort of set the stage for the newer analysis I'll show you. So this is, of course, is a review by now, especially after hearing the talks today. But as you know, World Trade Center responders were exposed to both fine and coarse particulate matter and other toxins in the days and months after 9/11. In addition, of course, to the psychiatric trauma from their surroundings. So this figure just shows some of the possible ways in which these types of exposures may have entered the lungs and brain, in addition to PTSD potentially affecting the brain through systemic inflammation. So this was sort of some of the initial hypotheses behind, perhaps mechanisms behind why the group was interested in looking at both cognitive and physical function in responders, because of course these are both affected by brain health. And in addition to the mechanisms just shown, we also have reason to believe from other cohorts and other studies that there may be cognitive and physical functional impairment in groups exposed to these types of exposures and to PTSD. So first, studies have found in other populations that other types of air pollution, like living near a major roadway, is associated with cognitive impairment. And studies have also found that PTSD from other types of traumatic events and in other populations have been associated with cognitive impairment, potentially through systemic inflammation. And we also know that cognitive functioning is associated with physical functioning, which is mostly what I'll focus on today. So by physical functioning, I essentially mean everyday tasks like sitting and standing from a chair, walking up and down stairs, activities of everyday living that are also sort of surrogates for overall health and function and independence. So this figure is from one of Dr. Klusten's papers, which is a review paper looking at different cohorts, different studies, not World Trade Center responders, just looking at the correlations between different physical functioning measures and cognitive impairment and decline. And what he found is that these measures of physical function were associated with cognitive impairment, both cross-sectionally and also where physical functioning at baseline predicted follow-up cognitive function. So these are definitely related, which makes sense because they're both indicators of biological aging. And of course, cognitive function and physical function decline in most people as we age. But if they decline earlier, this might be considered accelerated aging. And so this is one thing that we're worried about in this group. And then I just wanted to note that physical function is associated not only with cognitive function, but with other health outcomes as well, including cardiovascular disease, falls, frailty, mortality. So it's really, these are measures of overall health and potentially even thought of as biomarkers for health. And they can be pretty easily assessed using things like just testing speed at which someone can walk 12 feet, that's walk speed here, or gait speed, grip strength, which I'll talk more about, chair stands, sitting and standing up from a chair and being timed and converted to speed. So these are all measures of physical function. And then coming back to cognitive dysfunction for a moment and back to World Trade Center. As you have heard today, we have seen pretty concerning levels of mild cognitive impairment in this group. So these are just some estimates from the MoCA or Montreal Cognitive Assessment, both prevalence and incidence. And another study, again led by Dr. Klusten, found that when comparing the responders to age-matched controls from other studies, they also performed worse on these individual cognitive and motor tasks from the Cogstate Brief Battery, which is a computer-assisted set of game-like tasks, as you heard about earlier. So the figure here shows World Trade Center responders with PTSD all the way to the right. Those are the dark gray bars. The white bars being World Trade Center responders without PTSD, and the light gray bars being age-matched controls, or those who are not responders. And what he found was that in every age group and across these three different types of cognitive or motor function, like memory, reaction speed, and processing speed, the responders performed worse on all tasks compared to their counterparts who were not responders, and PTSD is highly associated with this lower function. So as I said in the beginning, this makes sense because some possible causes we talked about were PTSD and also exposure to the neurotoxic particulate matter. So both PTSD severity and these exposures are longer time on site, which is sort of a proxy for exposure, were both associated with mild cognitive impairment in these studies. But I'm mostly going to focus in on PTSD here. So as I said, cognitive function is related to physical function as well. And indeed, we have also seen physical functional impairment in this group. So this is a paper from Dr. Klooston's paper that looked at physical functional limitations. So on the left, you see a bar graph that shows these different measures of physical function from the short performance physical battery. So again, these are measuring things like walking speed, chair rise speed, et cetera. And what we see is that responders who, again, are all the way to the right, this time in gray bars, have much slower chair rise speed and walking speed than their counterparts or age-matched controls. And it is difficult to find good comparative data because, of course, many other studies don't start measuring these types of things until later in age. And we have a slightly younger cohort, so this is among responders aged 65 to 69. But this is indicative of possible functional limitations. And importantly, on the right, what you see is that those who have functional limitations, and this is based on a cutoff score from those measures on the left, from the SPPB, those with functional limitations had higher levels of PTSD over time as well. So once again, we're seeing that PTSD is associated with impairment, this time with physical impairment. And then I also just wanted to talk about grip strength specifically. So grip strength has been measured in the World Trade Center General Respondent Cohort since about 2015. And it's measured using something called a dynamometer, an example of which is shown here. And this ultimately measures upper body strength, but it really, again, is a surrogate for a lot of different measures. So it's been shown to predict all types of health outcomes, including mortality, and it's pretty robust to reverse causality, as has been shown in different studies, and seems to be robust against things like motivation day-to-day as well. So it really is a good and quick measure of sort of getting at overall muscle function and or lack of impairment, rather. So in our group, others have found that lower hand grip strength is associated with PTSD again. So in addition to those SPPB measures, which are mostly lower body extremity, and re-experiencing symptoms in particular seem to be highly predictive of lower hand grip strength. Another paper found that PTSD was linked to over 3 times the risk of comorbid cognitive and physical impairment, this time including the hand grip strength measure with the SPPB measures. And in fact, reduction in hand grip strength or lower, weaker hand grip strength was the most likely to be comorbid with cognitive impairment compared to the other physical impairment conditions. So I think the next question here was about timing. So we now know that PTSD is associated with cognitive impairment, with physical impairment. So which comes first? And so this last paper I'll show you before I get to the newer analysis by Pilecki and colleagues was looking at this question. So they found that it really seems like physical functional impairment seems to occur first in our group. So as I said, both of these tend to decline over time in aging populations, but in our group, they seem to be happening a little earlier, and physical functional impairment seems to be happening before cognitive functional or mild cognitive impairment. So this tells us that it might be really important to predict physical functional impairment as a early indication or first sign of further problems. So that's really the focus on physical function here and hand grip strength in particular, which has been shown to be a very good predictor of future physical problems. So then that all brings us to the current study where we have updated measures of all of these physical functional impairment measures, and in particular, hand grip strength. We have more data on that than the other measures. So I used the general responder cohort based in Long Island for this analysis. We had over 10,000 individuals contributing to almost 30,000 visits. So we have follow-up longitudinal data on the physical functional measures, and in particular, hand grip strength has the highest sample size because that test was able to be continued during the pandemic, while others were paused due to physical distancing. But we roughly have data between 2015 and 2023. Hand grip strength was measured with a dynamometer, which was digital and computer assisted. We also used the SPB measures I already told you about. And for the exposure of interest, I'm really focusing in on PTSD here. And so what I used for these particular analyses were PTSD or probable PTSD at visit 1, because I'm interested in potential long-term effects of baseline PTSD predicting potential long-term changes in physical function. So I used the PCL cutoff of 44, which is the DSM-4 suggested cutoff from visit 1. I used GEE models to adjust for repeated measures because we have individuals coming and contributing multiple waves of data, and adjusted for demographics, medical conditions, and exposure variables. And by exposure variables, I mean those that we were talking about earlier, like exposure to the dust cloud and time on site, to see whether PTSD is associated with physical function above these factors. So then just to show you a few key findings. First, just descriptively, I thought it might be interesting to show you what the hand grip strength looks like over time in this group. So on the x-axis here, you see age at visit, and on the y-axis, you see maximum hand grip strength in pounds. So out of 2 trials, maximum strength using the dynamometer. So as we would expect, hand grip strength is declining over time, which does make sense, again, with aging. But something I think worth pointing out here is that the mean grip strength is also relatively pretty low. So as I said earlier, it's hard to find data that really we can use to compare these responders to, but based on some data that is available for their age group, this mean, which is about 66 pounds in terms of hand grip strength, seems to be pretty low for that age group. So this is potentially concerning. And just in a simpler linear regression, 1 year increase in age is associated with about a 0.73 weaker hand grip strength, just to give you a sense of this relationship. And then in terms of PTSD, so this is showing you baseline PTSD as the exposure, and then 3 different outcomes: that maximum hand grip strength you just saw, walking speed, and chair rise speed. So these are all from the GEE models with the confounders listed at the bottom. The first column is just the crude beta estimates, so not adjusted for confounders. Second column is adjusted, but these betas are unstandardized. So these are just using the raw variables. So for example, those who met criteria for PTSD at baseline had over a 4-pound weaker hand grip strength compared to those who did not meet criteria for PTSD. So that is how to interpret these. And so what we see is that they are all negatively associated. They're all statistically significant. And there's, of course, different scales here. So hand grip strength has a lot more variability than walking speed or chair rise speed. So the numbers themselves look very different. But the relationships with each other are all pretty consistently negative. And of course, it's not the exact value per se that we're interested in terms of these types of measures, because they are surrogates for overall functioning and health. And then I also looked by gender, so I was just curious whether there might be any differences in these relationships by gender. So this shows you the same type of betas that I just showed you, but just graphed to sort of visually see whether there are differences. So the horizontal line is the null value here at 0, because these are beta values. And what we can see is there really doesn't seem to be much of a difference here. Of course, we have a lot fewer women in the responder group, so those confidence intervals are much wider. There's less precision, so that's why the vertical lines are so large for the women in particular. So it's a little hard to compare, but both among men and women, meeting criteria for PTSD is associated with weaker hand grip strength, and there does not seem to be effect modification by gender. But I'm curious to look at other types of potential effect modification as well in future analyses. And then finally, I was interested in potential differences across symptom clusters too. So of course, PTSD can have different types of symptoms like re-experiencing, which is usually nightmares, flashbacks, and that is often considered sort of the hallmark symptom of PTSD is the nightmares. And so I was really interested in whether that might drive some of this relationship, which we did see in the earlier work. So in the papers by Mukherjee and Diminish that I showed you earlier, they did find that certain PTSD clusters seem to be most associated with physical functional outcomes. So that is roughly consistent with what I found here. So this is showing you the 4 different symptom clusters on the DSM-4, what a beta value is for a 1-point average increase in the score for these subscales. So they're all, again, statistically significant. They're all negative and consistent, but I highlighted the 2 that are most predictive of weaker hand grip strength. So re-experiencing and emotional numbing. So there's definitely, I think, further work to be done here, but this tells us that these 2 clusters may be driving some of this relationship, and in particular, potentially nightmares. So I've looked at nightmares specifically too in a preliminary exploratory work that I don't have time to show here. So just to sum up what I've shown you here today and what I'm interested in doing in the future. So first, we so far have found pretty consistent longitudinal relationships between PTSD at baseline and then follow-up objective physical functioning measures. And so by objective, I mean the fact that prior work that has looked at PTSD and physical functioning has mostly relied on subjective measures. So asking patients how they feel they have been getting through their day, how they feel their muscle strength has declined, et cetera. So work has shown that individuals with PTSD may over-report on scales like that. So having tools that are more objective, like dynamometers or timing someone walking, is considered a better and more objective tool. So this is an improvement in prior work compared to other cohorts in that we do have these measures and we now have them longitudinally. And we also found that the relationships with PTSD remained statistically significant after adjusting for a range of confounders, including the World Trade Center physical exposures. So it really seems like PTSD is what's important here. So in the exploratory analyses, we found that re-experiencing symptoms and nightmares in particular may be what's driving much of this relationship. So this is important in thinking about potential mechanistic pathways. Maybe disruption in sleep is really on this pathway, and we need to be looking more at that because, of course, sleep is going to be associated with overall physical function as well and potentially cognitive function. And I think this is really important because, as I said in the beginning, physical impairment may be an early marker for eventual cognitive decline, in addition to other health outcomes. And then finally, our group has just been talking recently about whether it makes sense to come up with sort of a new diagnostic category here for what we're seeing. This is sort of adding to a body of work that has pretty consistently seen in the World Trade Center responders cognitive impairment, physical functional impairment related to PTSD that does not seem consistent with Alzheimer's disease, as you have also heard today, in terms of the imaging and other symptoms. So we think this is something important to think about going forward. And next steps for my own particular work, I'm interested in looking at those PTSD symptom clusters I showed you for the other SPPB outcomes, so not just hand grip strength, but things like balance and chair speed, to see if we can replicate those findings. I'm also interested in grip asymmetry or difference between grip strength in each hand, which can be considered a measure of dysfunction as well. I am curious whether there are potential mediators on this pathway. So I just mentioned disrupted sleep, but there may also be other health factors, medical factors, and behaviors that may be on the pathway between PTSD and physical function. And I'm also curious about looking more closely at the changes in PTSD and physical function over time together. So today I just showed the baseline PTSD and follow-up physical function. But I'm now starting to think about time-varying PTSD and how, if PTSD, for example, gets better in some over time, will physical function improve or does it still incline? And how do those two variables sort of co-vary? And then finally, interested in how these relationships might change based on different demographic groups, and potentially vulnerable subgroups. So that is all. Just would like to acknowledge the funding, mentors, collaborators, those who provided the papers early on that I showed you, and also, of course, the responders who gave their data.
So, my question again is, are we at a point where we would recommend physical assessment functions for routine medical care for people at risk? Are we there yet, or we're not there yet?
So personally, I think that it makes a lot of sense to include physical functional screening in clinician visits. Of course, time permitting. I know it's always difficult with clinical visits in terms of adding more things, but these types of measures are actually pretty easy to administer, quick, and they're highly predictive of all kinds of outcomes. So not just cognitive impairment, but as I said, cardiovascular disease, falls, mortality. So I do think it makes sense to do this, especially at high-risk patients who might be undergoing different surgeries or something. And also important to think about for a patient with PTSD, because we already, there already may be challenges in treating PTSD. So if we know something's going on with the brain potentially, and movement, and there might be atrophying in the brain, then this is, of course, relevant for PTSD treatment. And there also may be different interventions we might be able to do on physical functioning, like progressive muscle strengthening. And so I do think it's worth pushing for that.
Steve Koza, Uniformed Services University, Bethesda, Maryland. Great presentation. Thank you very much. I was wondering, did you happen to have any alcohol use data to look at as it related to some of these outcomes, particularly in the physical functioning?
Yeah, that's a great question. I have not looked at that yet, but that is on my list of things. And in particular with this analysis I showed you here, I was most interested in baseline PTSD. So I think we have some more information on that later in the follow-up, but I definitely want to see whether that's associated.
Yeah, thanks.
Any other questions from the audience? Thank you very much.
Thank you.
Very good. Our next speaker is Monica Vashtuk. She's an associate professor of psychology at Rosalind Franklin University of Medicine and Science. She completed her undergraduate training at the Department of Experimental Psychology in the University of Oxford in the UK. She received her Master's in Science, a PhD in Behavioral Genetics from King's College in London. And following her graduate training, Dr. Vashtuk joined the Department of Psychiatry at Stony Brook University in New York. Thank you.
Hi, good afternoon, everyone. Thank you so much for this kind introduction. I'm very excited to be here. I am currently based in Chicago, but I have been working with the World Trade Center responders team and data at Stony Brook for about 8 years now. So you might have seen me at some of the previous meetings. And I'll be talking today about one of the projects that I'm most passionate about and most excited, and that's the work that I've been doing closely with Dr. Laft and Dr. Kodov, looking at genetic markers and genetic vulnerabilities in 9/11 responders, with the broader kind of goal of trying to understand whether we can predict some of the long-term health outcomes using genetic predictors. So I'll be talking about some of the papers and work we've done in the past, and also some of the ideas for future work in clinical translation as well. And I'll be mentioning work done by many people who are already in this room, because this has been truly a collaborative effort. All right, so that is the talk. All right, so one of the main motivations for looking at genetics is that it's one of the biomarkers that doesn't change at all over time, so it's pretty stable. So it can be a pretty good predictor way before any onset of disease. We are also I kind of came up with this project. We were at a good point where there was a lot of data that allowed construction of polygenic risk scores. So I'll be referring quite often to polygenic risk scores, which are basically an aggregate scores of multiple genetic variants associated with disease risk. So you can basically create a polygenic risk score and use it for multiple research and potential clinical purposes as well. So our kind of biggest goal was to identify patient populations with the greatest intervention needs within the World Trade Center community. And some of the kind of outcomes that are relevant for prediction include some of the chronic treatment-resistant mental health conditions, but also physical illnesses such as cardiovascular or cancer diseases. And kind of like based on some of the previous work that has been discussed today, there's also a lot of interest and need to predict cognitive decline, maybe some of the MCI and dementia outcomes as well. And we're pretty cognizant that in addition to informing the World Trade Center responder program, some of the work and the findings would also translate pretty well to other occupational cohorts, as well as veteran military populations as well. So the kind of broader goal is to really reduce the burden of PTSD, but also other disorders, by identifying people who are kind of at risk, maybe early on or prior to exposure, and would benefit from interventions even before the onset, in terms of like resilience training or psychoeducation or just screenings. So I will be talking largely about PTSD research first, and then also kind of comment on some of the findings in other domains as well. And in terms of the polygenic risk scores that I will be talking about, there's a pretty kind of wide range of risk scores that can be constructed and used. And the prediction is like never really domain specific. So you will see that we use polygenic risk scores for other psychiatric conditions, for other kind of mental health, physical conditions as well. So basically just use whatever tool kind of worked best in terms of prediction. All right, so starting from some of the earliest kind of papers and work that we've done, this is one of the first papers that we published based on like the very first kind of batch of data that we genotyped. So we had close to, for this particular analysis, close to 1,000 participants, about 220 PTSD cases, and the rest of individuals were trauma-exposed controls. And you can see that we basically found that polygenic risk score predicted PTSD diagnosis. So on the x-axis, we have just the highest quartile groups for the depression polygenic risk score, and the odds ratios are on the y-axis for the diagnosis risk. In the same paper, we reported that polygenic risk scores also predict PTSD symptom severity. So we had a variety of polygenic risk scores that we tested. Again, the reference group is the lowest PRS quartile. And as the genetic vulnerability to various psychiatric conditions increased, we also saw that the risk for PTSD severity increased as well. The effect sizes are standardized, they're pretty low, but just want to highlight that we did adjust for quite a lot of demographic variables here. We also adjusted for the trauma exposure severity, so this is kind of independent of trauma exposure. There are multiple kind of color bar here, is just because we looked at overall PTSD symptom severity, but we also broke them down by the symptom clusters as well. But the pattern is roughly the same. So now we have a much larger sample that's genotyped. Almost everybody who was eligible has been genotyped. And we kind of report very similar findings at this point as well. So for the odds ratios for PTSD diagnosis, various polygenic risk scores give us ratios of about 1.13 all the way to 1.3. And again, in terms of associations with the dimensional PTSD symptoms on PCL, we see the betas of around 0.1 for various polygenic risk scores. So these are kind of prediction that's independent of the demographic predictors that we know at baseline, and also trauma exposure severity. In fact, trauma exposure severity kind of seems to be a very independent vulnerability factor from the genetics. So we kind of are operating within this additive model where we have genetic vulnerability kind of adding on to the environmental exposure. So this was kind of like cross-sectional. Even the kind of severity level was very much average, about using kind of average scores of the data that's available to us. And the diagnosis, kind of lifetime PTSD diagnosis in the population. But we're also able to do some of the longitudinal work as well. So in the very first paper, in the smaller sample, we were just split our population into individuals who had like the healthy trajectory, that's majority of participants, and about 17% of participants had the severe trajectory. That kind of went above the probable PTSD cutoff for diagnosis. So here, the kind of dependent variable was just the trajectory class. And again, we found that polygenic risk scores predicted the trajectory class. So again, I think one of the very first papers in PTSD literature that looked at that prediction longitudinally as well. Dr. Mann, Frank, who presented earlier, has done this work now in the full population that has been genotyped, so much larger sample size. He's been able to also model the trajectories, not just as classes, but instead he's modeled them like for within person trajectories, so much more personalized. And in the top panels, you can see that, again, polygenic risk scores are predicting the trajectories pretty well, with the highest polygenic load being associated with the more severe trajectory. And then just for comparison, we also have models where we used the trauma exposure severity as a predictor of the trajectories as well. And you can see that, again, we have the separation of severity of individual within person PTSD trajectories depending on the exposure. So in addition to kind of looking at the risk factors as environmental risk factors kind of additive to genetics, we can also think about different ways in which genetic vulnerability can potentially be modified or moderated by various environmental or social variables. So I was working with my graduate student on some of this data for her dissertation. Basically, we're very interested whether the social support that participants reported close to 9/11 during the first visit was in any way kind of affecting the prediction, the polygenic prediction of PTSD. And we found that having at least one supportive coworker during 9/11 actually buffered the association between the polygenic risk score and PTSD severity. And we did not see this kind of buffering effect looking at family social support. So kind of very preliminary findings. This is unpublished work. This is just dissertation work. But kind of gives us a bit of an idea that maybe there are ways in which different environmental kind of protective factors might buffer some of the vulnerabilities that people inherited. And just the final slide on the PTSD genetics specifically, we were fortunate to also contribute some of our data to the Psychiatric Genetic Consortium collaboratively. So this is one of the largest, I think this is the largest study that has basically combined multiple data sets of PTSD patients, including the Million Veteran Program, in order to have the kind of largest data set for discovery of genetics that are associated with PTSD. And this work that has been recently published and kind of includes some of members of our cohort and has found 95 individual kind of SNPs associated with PTSD status. 80 of those reported in this new paper are new. So the field is really moving forward. This is a very kind of exciting area. And again, like in addition to all the kind of impact that the research might potentially lead to in the 9/11 responders population, this is kind of like work that also contributes more broadly to the research field. As we were genotyping participants in the study kind of ran very close to the COVID-19 pandemic, we were just very interested in exploring prediction of other phenotypes, other outcomes in this population. So this is one study that we conducted, just looking at genetic prediction of COVID-19 severity. So again, a bit smaller population here, over 800 responders who had a confirmed infection quite early on in the pandemic. And we're just very interested whether various polygenic risk scores might be associated with the COVID-19 severity. And the outcome variable was like a pretty severe COVID-19 outcome with like hospitalization included. And we found some really reasonable odds ratios for asthma PRS as well as allergic disease PRS. Some of the early polygenic risk scores that emerged for COVID-19 have also been associated with COVID-19 severity in 9/11 responders. Again, these associations adjusted for demographics as well as the lower and upper respiratory symptoms, just to make sure that we're not just capturing vulnerability to asthma or other respiratory conditions. Some of the other studies that kind of recently came out from this work, again, a study conducted by Dr. Mann, has looked at polygenic risk scores in addition to multiple other predictors of cognitive impairment outcomes. Here's kind of vulnerability to mild cognitive impairment. And the polygenic risk score for Alzheimer's disease seems to be predicting the odds ratios for mild cognitive impairment. And likewise, genetic kind of predisposition to have a higher educational attainment seems to be slightly protective against mild cognitive impairment. Another kind of target phenotype kind of condition that we've published on and been interested in is the chronic kidney disease. Again, we find associations between the polygenic risk score and the more advanced baseline kidney stage, again, adjusting for covariates. So kind of once you have a kind of genetic information on participants, you can really explore various kind of outcomes, because once you have the genotype information, you can pretty much compute any polygenic risk score that interests you and has been developed previously. So in terms of the summary of empirical findings, just to kind of run through everything that I've mentioned just now, we have demonstrated that we can use this genetic information to predict PTSD severity and also onset following trauma, and some of the predictions are pretty long-term. Some of the longest longitudinal studies have been spanning about 20 years of data currently. There's potential prediction of MCI, but not dementia. That's probably because the population is still a little too early on average in terms of their age for dementia prediction to come through. It might be something that shows up in the future. We had some success with the kidney disease outcomes and some of the COVID-19 severity symptoms. Although they mentioned that the long COVID kind of symptoms were a little harder to predict genetically, so that one we couldn't really capture. And this is just the polygenic risk score prediction. Obviously, there is all of these kind of outcomes are multifactorial. They have very complex etiologies, and genetics is only one small part of the entire equation. So there's also interesting kind of insights that we might be getting from all the environmental variables that we have as well. So the trauma severity and other environmental factors seem to be additive in addition to genetics. So we might be able to kind of increase prediction and maximize some of the algorithms by adding more kind of exposure and other variables, so they're not like redundant with each other. Social support seems to be kind of a good predictor of PTSD, but in addition, it might buffer genetic risk. Again, more application and more work is needed in this area. And we haven't really done that much work yet in biomarkers, but that seems to be one of the kind of more mechanistic and exciting areas of future research, is to look at some of the pathways from genetic vulnerability to PTSD and other outcomes, and trying to understand whether they come from maybe inflammatory markers or proteomics, or there might be some changes in metabolic markers that are associated with polygenic risk score. So some of these biomarkers might potentially inform a little bit more of the mechanistic story, as opposed to just focusing on prediction. In terms of the clinical translation, polygenic risk scores are not really clinically used. At this point, there's a lot of commercial uses of polygenic risk scores where people can kind of run a genetic test and receive some of the results for themselves. There is a lot of clinical validity to polygenic risk scores, but that we don't really have like randomized controlled trials that really show that integrating polygenic risk scores is better than the care as usual. So I think it's kind of at the verge of clinical translation, but it's not there yet. The prediction is very small. Ideally, you'd want to kind of have validated and replicated algorithms that have a bit more than just genetic information in them. But there are really exciting opportunities for critical translation and research, and also clinical research more broadly. As I mentioned, there is kind of like a really, it would basically mostly serve a purpose as a screening tool to kind of identify people or groups of participants at risk, or also individuals who might be extra resilient. That might also be pretty informative. As I mentioned, it might be pretty useful in the future to combine polygenic risk scores with other risk factors in clinical populations. And one of the advantages is this long-term prediction. So kind of like knowing a lot of baseline information like demographic characteristics and trauma exposure severity might be bolstered in terms of prediction if you also know people's kind of underlying genetic vulnerability as well. Also for clinical translation, I think the cost effectiveness of polygenic risk scores is going to be a pretty good advantage. It's not really that expensive to genotype at this point, a lot of people have already been genotyped just on their own, out of their own interest. So that kind of data is going to be probably much more affordable than a lot of other biomarkers. In terms of future needs and kind of thinking about the future directions, I think in general, larger sample sizes for other modalities would be helpful at this point. I think we have a pretty large genotype population at Stony Brook, and I think it's, at this point, I think the most interesting questions really pertain kind of connecting the genetic data with all the other sorts of data. So of course, the biological modalities that I just mentioned earlier, in larger sample sizes would be really kind of helpful for developing some of those mechanistic explanations of like, why do we have this genetic prediction? Is there any specific pathway that mediates the associations? But in terms of the phenotyping, I think there's a lot of other kind of interesting phenotypes that would matter as well. So in terms of comorbid disorders, anxiety, substance use, pain, insomnia, I think being able to see if you can predict this kind of outcomes in 9/11 responders would be really interesting. There are behavioral determinants of health as well as social determinants of health that can be combined with polygenic risk scores. Again, maybe in additive models, maybe as moderators, but there's a lot of really cool ideas and interesting questions that can be asked if we had like that kind of deeper behavioral and social data. And again, knowing that the population is aging and there's a lot of really important questions about MCI and dementia, I think gathering more fine-tuned aging-related phenotypes, in addition to obviously the cognition itself, but also functioning, maybe potential healthcare utilization, maybe caregiving, receiving care, might also kind of make those models much more complicated. And kind of better reflecting the reality of the kind of like really multi-dimensional prediction of health outcomes. All right, so I just wanted to thank everybody for collaborating on this work, and I'll be happy to answer any questions.
Questions?
Yong Zhao from NYU. Okay, very basic question. When you say polygenic risk score, are you using SNP genotypes or you use SNPs? Okay.
So these are SNPs in aggregate across the entire DNA. They're weighted by the importance that they play in each phenotype.
Right. So for example, when you say you predict PTSD, right, trajectory or score, right? So did you use a linear regression, use a PCL score to form the polygenic score, or you use a cutoff 44%, use a logistic regression? And what kind of polygenic risk score? There are many different ways, right?
So the polygenic risk score is the predictor, right? So we run logistic regressions for the class, the trajectory classes. Some of the individual kind of within person trajectories were more like multi-level modeling. But the polygenic risk score itself is literally just like a single variable in the model. It's kind of like a combination of all the different SNPs. They kind of added up to create like person's kind of risk level from, I guess they were standardized, but like from 0 to 1, you could say.
So the polygenic risk score, you got it from exposed and unexposed? In other words, how did you get it?
Yeah, that's a good question. Most of the polygenic risk scores come from case control studies.
Yeah.
So there will be like a really huge study that has hundreds and thousands of cases, hundreds of thousands of controls, and that's usually the equation, that's usually the discovery sample that causes the equation. Right. Some of the polygenic risk scores can also be constructed using dimensional phenotypes. I think some of the neuroticism and educational attainment, that kind of phenotypes would usually be used dimensionally to create polygenic risk score. But for some of the main kind of PTSD, depression, polygenic risk scores, these are based on categorical case control definitions.
I see.
Yeah.
I see.
So yeah, I had a limit of slides, so I couldn't really put like methodological slide explaining how you create a polygenic risk score. But that's a really good question. I think there are efforts now to also create polygenic risk scores for PTSD using like dimensional definition of PTSD as well.
Right. So you can further look at PTSD clusters, right? So the 4 different clusters, things like that.
Yeah, so we looked at them as outcomes, but we also had some information about PTSD clusters as from polygenic risk scores as well.
Okay, thank you. Thank you very much.
Other questions? Do you think the size of your cohort limits your predictive ability for clinical translation? And I appreciate the slide, by the way.
Some of the first, the batch one results were kind of limited in power. Also, the COVID analysis were limited to participants who had confirmed COVID, so they were smaller, on a smaller side. I think the full genotype sample is pretty big for that kind of tests. So I think at this point we've achieved a power that allows us to detect pretty small effects. So I think we're doing okay at this point, yeah, as a total cohort. Yeah.
Any other questions from the audience? Thank you. Thanks very much.
Thank you so much.
All right.